Hello, everyone, and welcome to this session of the Porous Media Tea Time Talks. My name is Marcel Moura, and I am joined here at the studio by my friends Mohammed Nureipur and Chiara Hekalkati. And together with the whole team of the Porous Media Tea Time Talks, we would like to welcome you to this session. This is session number 57 of the PMTTT. And today we have two speakers, Saeid Ataei from the University of Edinburgh and Yao Xu from the University of Oslo. And we will start straight away. And we would like to thank the Interpor Young Academy and the Interpor in general for partnering with us in this little adventure of ours. Okay, so we'll start with Saeid Ataei. And he is a research associate in energy at the University of Edinburgh School of Geosciences. He earned his PhD from Heriot-Watt University, specializing in fluid flow through porous media for geological gas storage and enhanced hydrocarbon recovery. Saeid has significant experience in collaborating with industry stakeholders such as Woodside Energy, Total, Wintershall, BP, among others. And he's going to talk to us today about the cyclic flow characteristics of sandstones during geological hydrogen storage in saline aquifers. So, Saeid, your presentation is on, and the floor is yours.
I'm at the University of Edinburgh, and today I'm just going to talk about cyclic flow characteristics of mainly sandstone reservoir during geological hydrogen storage in saline aquifers. So here we are I'm just going to share the experimental findings which I have performed during the last couple of years at the School of Geoscience. So first, I will go through the objective and background of this line of research. It will be followed by the experimental program, and then some results and some brief results and discussion will come after. And finally, we will go through the conclusion of this line of research. So, as you probably know, recently it has been shown the geological hydrogen storage is one of the main potential for storing hydrogen during the low demand season, and the porous rock can also be used for just producing hydrogen during the high demand season. But the problem in this regard, the problem with storing hydrogen in porous rocks, storing gas in porous rock, is just due to the capillary forces. There is always the chance of residual trapping and also the capillary trapping of the gas. So this reduces the efficiency of the system. So here at the School of Geoscience, University of Edinburgh, we try to mainly perform experiment and simulate the condition of each reservoir and perform experiment using different rock types to measure the residual trapping for different rock types and also investigate the impact of rock type on the residual trapping. So as I mentioned, the aim and objective of this line of research is to evaluate and measure the residual trapping during cyclic flow of hydrogen and brine onto different rock types and measure it for different rock types, and by comparison of the residual trapping for different rock types, we would be able to compare and get the comparison between the residual trapping among different rock types and study the impact of rock types on residual trapping. And also, in addition to those, we would obtain invaluable experimental data sets for reservoir simulation and reservoir modeling. So this study, this line of research has started a couple of years ago at the School of Geoscience by my colleagues, and they performed some XR, some microcity analysis on the smaller scale core sample, and they realized that on the water wet system, on the water wet rocks, the snap-off mechanism is one of the active mechanism of residual trapping of the hydrogen. So for further detail, you can go through the reference and just read the detail of the work performed at the School of Geoscience. But to build up our understanding and also to perform a quantitative analysis of residual trapping, currently we are performing core flooding experiment on the longer core samples because the problem with the experiments, a microcity experiment on the smaller scale core sample, they have got some sort of experimental artifacts, mainly capillary end effects, because the boundary is really small in the smaller scale core sample, and the capillary end effect can impact the experimental findings. So although the microcity can provide invaluable findings in regards to the mechanistic studies of the residual trapping, but they are not the best option for qualitative analysis of quantitative analysis of the residual trapping. So at the moment, we are just performing experiment using longer core sample, larger core sample with mainly core sample with diameter of 1.5 inch in diameter to investigate the impact of residual trapping. So before each experiment, there is a methodology, and each rock sample would go through the core taking process, and the core sample from different reservoir rocks would be taken in our workshop here. Our enhanced methodology would be applied to them, and then after that, each core would get cleaned, and then it would get loaded to the core holder system, and a special care is just taken in this state to make sure there is no connection between the overburden fluids and also the pore fluids. And then after that, for each core sample, the basic properties of each core sample would be measured, which is effective porosity and also absolute permeability. And also, finally, the sample selection would be conducted by comparing the measured basic properties with the values measured or obtained by the well logging data. So a special care is taken to make sure the core is representative of the reservoir rock. And then after that, the cyclic flow experiment would be conducted at a specific reservoir condition to investigate the fluid flow characteristics. So here in this slide, I'm going to talk about experiment performed on two distinct core samples, sample number 1 and sample number 2. As you can see, these two graphs shows the fluid saturation at different stage of at different pore volume of injected of fluids, and a stage of the cyclic flow experiment performed at high pressure and high temperature on sample number 1 and sample number 2. The blue curve is related to the water saturation, and the red curve is related to the gas saturation. So as you can see, both of the experiment are started once the water saturation was 1. So this shows that the experiment started from the first drainage process, and so as you can see, the first cycle of the gas, each cycle was continued until a plateau was reached on the saturation profile and also the differential pressure across the core sample, which I didn't bring here due to the limited of the time. And then it should be mentioned that at each stage of the experiment, to make sure there is no capillary end effects, the bomb flooding was conducted. So as you can see in sample number 1, the first gas cycle resulted in an initial gas saturation of 0.6. But in sample number 2, the initial gas saturation for the gas cycle 1 is around 0.53. And so once the steady state condition was achieved for both core sample, they were subject to the first cycle of water, and the first cycle of water resulted in the residual trapping of around 0.32 for the sample number 1 and around 0.23 for the sample number 2. And the experiment for both of the sample was continued for the second cycle of gas, second cycle of water, and third cycle of the gas and water to investigate the impact of multiple cycle on the residual trapping. So several important features of the finding should be highlighted here. So the first finding, if you can take a look at the residual trapping, so the red graph, it seems that the residual trapping remained constant after the first cycle for both of the sample. This is a great finding, at least for these two samples. This shows that we can just use the the data for the first cycle to just simulate the following cycle of during the hydrogen storage. But this is just related to these two samples. And at the moment, we are just performing more experiment on more different rock types to just confirm this observation and see what caused that impact on the experimental data. And also, as I mentioned, the sample number 1 has higher initial gas saturation as compared to the sample number 2. And also, it has got higher residual gas saturation as compared to the sample number 2. So, different flow characteristic was observed for the sample number 1 and sample number 2. So also to further investigate the impact of rock type, the mercury injection capillary pressure test was also conducted on both of the sample, and it seems that the larger the pore sites in the sample number 1 are larger than the sample number 2, and so this can be because the wettability of the system in both of the sample, because it's a saline aquifer, is water wet, and gas in both of the samples tends to move in the middle of the larger pore. So because in the sample number 1 there is larger core samples, so this might be the case. We got the higher initial gas saturation. And also, as I mentioned, because it's just a water wet, so the snap-off mechanism is the main, could be the main trapping mechanism. And so the larger size of the sample number 1 caused more snap-off mechanism, more pronounced snap-off mechanism, and the more residual trapping in the sample number 1. And also the results clearly shows that there is a difference. There is a clear difference in terms of between those two samples in terms of the flow characteristics and also the MICP test results. So based on what I presented here, this is a result of a comprehensive study. So I just briefly mentioned what we are just doing here. So based on what we have observed through the previous couple of presentations, so it seems that the residual trapping remained constant after the first cycle for the presented results for these two core samples here. So at the moment, we are just trying to perform more analysis by performing experiment on more rock types and performing pore size distribution studies and also miscible tracer tests to see what caused that impact. And also, as I mentioned, different flow characteristics was observed for at least these two rock types. And it seems from this line of research for a specific reservoir, for each specific reservoir, experimental studies are required before proceeding with any field plan. So that's it. That's me. Thank you so much, everyone.
Thank you, Saeid. Thank you for sharing.
I hope I'm on time.
Yes, that was great. Thank you for sharing your experimental observation with us. Before I get the comments and questions from the chat, I will start with the question of my own.
Okay.
Given your experiences in the lab and several years of work in this line of research, what do you think? What is the basic difference when it comes to underground hydrogen storage compared to CO2 storage, compared to gas or methane storage? What is the principal difference when it comes to hydrogen?
So in terms of if you are comparing the hydrogen at the moment with just the natural gas, I mean the methane. So the natural gas, because the fluid flow through the porous media is mainly dependent on three main forces: the capillary forces, viscosity forces, and also gravity forces. So in terms of, because the hydrogen seems to be non-reactive at the temperature below 100, so there seems to be no significant difference in terms of the fluid flow through the porous media based on what we have observed between the hydrogen and also the natural gas, the methane. But in terms of the difference between the hydrogen and CO2, because CO2 is reactive with different rock types, so and also the IFT between CO2 and brine is quite different than the hydrogen and brine. So the fluid flow through the porous media is just different than between the hydrogen brine and CO2 and brine. So that's it, I guess.
Okay, great. And when it comes to recovery of the hydrogen, when we use saline aquifers or subsurface environments, porous media, as a battery to store and extract hydrogen or recover hydrogen, what do you think? What is the average number, a ballpark number for the sandstone reservoirs? How much of it can be recovered and how much will be lost as a residual trapped gas?
So the main idea of performing this line of research at the moment, because we are just performing experiment to understand the quantity of the residual trapping through different sandstone. And so at the moment, it's quite depending on the pore size distribution, and several factors would affect that. So the reservoir condition and also the degree of heterogeneity of the core sample. But you can just observe, for example, the residual trapping between 0.015 to the residual trapping of 0.4 into the sandstone reservoir. So at the moment, we are just showing that for each specific reservoir, the number is just quite different. And we try to see if we can just translate the result and get a general equation for the residual trapping, to see if there is any general equation for that or not. So, and the next stage here, so when we realize, for example, for each reservoir rock, for example, what is the quantity of the residual trapping, so the next stage, I think, would be if there is any method to just optimize that, to just reduce that amount of residual trapping or not. So at the moment, we try to understand that.
Excellent. Thank you very much. Do we have any question from the studio?
Yeah, I have a quick one. Very nice to say it. Thanks a lot. So you mentioned the effects of a capillary end effects, right?
Yes.
In both samples. Were they more pronounced in one sample than the other? And do you have like any kind of like a hint or like a general idea that, okay, if a sample is like this, and that, then capillary end effects are very important or not? Is there like any general description, or is it like a try each one and see if it's there or not?
Yeah, so the ideal case here, because I cannot see you, the ideal case here is because the capillary end effect is mainly dependent on the capillary forces. So it's just mainly created once at the outlet of the core, once you have a sudden transition from a medium with a maximum of capillary forces to a medium without any capillary forces. So it's mainly dependent on the capillary forces. So in the ideal case, so the core with the maximum capillary forces would just experience the more capillary end effects. But here we would perform experiments, and we try to have a just unique procedure to investigate the degree of the capillary end effect for each core sample, and this can indirectly shows also the degree of the capillary forces through the porous media. I hope I just answered your question based on what
Thank you.
we were looking for.
Excellent. Thank you very much, Saeid. I guess now we can invite our second speaker. Our second speaker of today is Yao. He's a postdoctoral fellow at the PoreLab Center at the University of Oslo. Yao did his PhD with a focus on reactive fluid flow in porous media. He is now working on the CO2 basalt project, where he is collaborating with other geophysicists and geologists to investigate CO2 storage potential in basalt layers. In this project, he is trying to experimentally elucidate the density-driven convective of CO2 in water system. Thank you very much, Yao, for joining us today, and the floor is yours.
Thank you very much, Mohammed, for the nice introductions, and thanks very much for having me. My name is Yao. I'm a PhD student of the University of Oslo. Today, I'm going to talk a little bit about my PhD project. And so I'm collaborating with some of the other geologists and geologists to investigate about the storage, the CO2 storage potentials in the basalt rocks. And we are working on different scales. So if you are interested in the geological carbon storage, you are very, very welcome to follow us on the Instagram. And we are trying to post some interesting things from time to time. And the geological carbon storage is being recognized as one of the most important or effective ways to reduce the anthropogenic carbon emissions in the atmosphere. And just like Saeid mentioned, there are many different kinds of mechanisms during the CO2 storage, among which solubility trapping is one of the very significant ways to store CO2. So what is the solubility trapping? Is where we try to dissolve CO2 into the water. So the CO2 dissolves into the water and it gets stored. Previous studies have shown like the convections could help to boost the mass transfer of the CO2 in the liquids. And so what is the convections? There are different kinds of the convections, and for example, the thermal convections and the density-driven convections. Today, we will only focus on the density-driven convections. So what is the density-driven convections? When CO2 gets dissolved into the water, because of its higher molecular weight, so it will make the liquid, which is mixed with CO2, have the higher density. So the heavier fluids will try to sink down in the formations, while the lighter fluid, which is not mixed with CO2, will float up because it has a lower density compared with the fluid that is mixed with CO2. There are different mechanisms in physical and chemicals behind these CO2 convections, and they are coupled with some. It is coupled with some other things such as the CO2 diffusions from the gas phases into the liquid phases because of the chemical potentials, and also there will also be the dispersions of the CO2 and in the liquids as per the different properties of the porous mediums. So these kind of mechanisms are coupled with each other and make the CO2 convections it's more difficult to understand. So our goal is to characterize these kind of convections, and we try to further investigate whether there's any correlations between the convections and the total amount of the CO2 that we can store in the liquid. And so our aims is not our goal is not only to quantitatively and to see about the so where is the CO2 migrate, but it's also to establish some of the effective mathematics models or physics models and to see the correlations. So here is the method we are using. So we are using the helixer cells and for the experiments. And this helixer cells is basically two glass plates that is bonding with each other by the gasket. And here we are taking advantage of the characteristics. Is CO2 is a little bit acid. So when it gets dissolved into the water, it will try to lower its pH. So by taking advantage of these characteristics, we try to add a little bit pH indicators into the liquid. So when CO2 gets dissolved, it will change its colors. And according to these colors change, and it will tell us much more information about how much CO2 is stored or where is the CO2 convection pathways. Here's we use the tailored pH indicators, which are 3 of them instead of just a single one. So the reasons of using 3 of them is just because if we just use one of them, you can see like it can just capture the range from a very small range, for example, 4 to 6 by the method rate. So, for example, when CO2, when this injection process just started, the brine water may be a little bit basic or maybe a little bit neutral, so it cannot capture the whole range of its pH change from 7 to 6. So that's the reasons why we put a bunch of the pH indicators, so that it will capture any minor or subtle change of the pH when even a small amount of the CO2 get dissolved inside. So then, say we have the helixer cells, we put it onto the ranks that we designed, and the black board, the white board is the backlight illumination. It could help us to provide the constant and the light sources. And then we put our helical sails on that, and then there's a camera which is aligned with the helical sails, which can just capture about the images at a regulated time. So that is the raw images we can get it, and it will show about where the CO2 convected inside of this liquid. So they have different kinds of the plumes. But the thing is like, how can we get more of the information from these raw images, and how can we take advantage of the image processing methods and to get more quantitatively things from that? So as for the image processing, of course, there are always lots of the pre-processing things, such as the perspective transform, because our camera is not exactly aligned with the helical sails. So there should be some ways to adjust it, and also the edge masking, which help us to get noisy, and then to just distinguish the liquid phase that we wanted to investigate. And then there's some of the more serious image processing ways, and we try to show about its pH and things. Then, just as I mentioned, the CO2 is a little bit acid, so it will just change the colors by its different amount of dissolved CO2. And for each of the pictures, and we will have bunches or millions of the pixels for each of the pictures. So for example, you can imagine like here, each grid is one single pixel, and the pixels will just the pixels will provide the details of the object at this time point and this point. And so if we take one of the pixels out, and for examples, and the color of it can be decomposed with the 3 color sources, which is red, green, and blue. Because here we use the 8-bit pictures, so each of the color components is ranging from 0 to 2 to 8 power 8, which is 0 to 256. So take one of the pixel, for example, and it's because it's very greenish, so it has a higher volumes of the green volumes, but it has a low volumes on both red and blue. So if we project the RGB values for the whole pictures into the Cartesian coordinates, that is what we get for that. And so the X and YZ represents about the red and blue and green values respectively, and the brightness is the square root of the sum of each of the color components. Here we find a very interesting things. It's like the brightness, which is the value I have shown here, will not exactly influence the pH. So what does it mean? So which means, like, imagine here we have a coordinate, and we have a lines OP. So if there are 2 points that is located on the OP, it will have the same azimuth and polar angles, which is theta and phi. And although these 2 points on the OP lines have different radians, but it will have exactly the same pH. So that's the reasons. For example, take 2 points. For example, if there's 1 point which is 2.45 and 1.8.82, you'll find like they are just this 2 times scale, and they have the same 2 angles, which is the theta and phi values, but they have different radians of that. But they will have the same pH. So that's the motivation. So how we change from the Cartesian coordinate into the polar coordinates. Here, for the polar coordinates, we also have 3 parameters. Phi, which is the radius, theta, though it's 2 angles. Just as I mentioned, because the brightness is not important anymore, so we just remove it, and we only care about these 2 angles. And these 2 angles is easy to calculate it by the Pythagorean theories of R and RGB volumes. And the black lines is the collaboration curves, and which we used to interpolate the things. And So how did we get the collaboration curve? And so we did a similar experiment, but the difference is like here, we saturated the Saeid cells with the solution that is with known pH, and then we titrate with the pH indicators, and then it has the uniform color showing, and then we capture it by the camera, and we get about the RGB volumes, and we calculate about the theta and phi. And because the pH is known, so we get the points, and we repeat the experiments, so we can get a bunch of the points, and we can get our collaboration curves. So for example, then for the certain experiments, we don't know the pH. And so what we did is like, okay, for example, for this point, and we just try to find its shortest distance to the collaboration curves. And then according to its intersections to the intersection, according to its intersection points on the lines, and we can find its pH of it. So then we can get a pH. For example, this is our Hilliard cells, and this is the pH field in space at a certain time step. pH is very important for the following investigation about the geochemical reactions and so on. But maybe we can do more things from that. Because we know that the pH actually is the showing of the total of the dissolved CO2 at the points or at the space. Is there any ways we can further calculate about the dissolved CO2 amount just by the pH volumes or from the hydrogen volumes? So that's the things that is how we did it. So here's we will show like the CO2 dissolves into the liquid is a chemical equilibrium process, and it's the instantaneous chemical equilibrium. It's instantaneous, which means like it reaches equilibrium instantaneously. So there's no kinetics in the reaction rate behind that. So here we can use the chemical equilibrium consistency and for each of the reactions, and there's a mass and ions balance and self-ionization theory, which is the hydrogen amount and the concentration of the hydrogens and the hydrogen dioxide is equal to 10 to minus 14. And then according to these kind of things, and we can get about our concentration from the hydrogen amount. The hydrogen is exactly what we talked about, pH. And because this kind of chemical equilibrium is known, what I have shown here, so then we can convert our pH field into our concentration field. And then we can know how much CO2 get dissolved into the cells or into the porous mediums totally. So here I have a bunch of videos I want to show. So this is the pH, this is the pH field. So when time changes, how it will change is pH in the Hilliard cells. So we inject the CO2 from the top. That is, so that is then here is the gas and the liquid interface. So if you can see it again, you can see when more CO2 get dissolved, and the heavier fluids or the fluids mixed with CO2 will gradually sink down until it reaches the bottoms and a little bit float up and mingles. Yes. And then so this is the carbon concentration field we constructed from the pH field. And the right panel is about the total dissolved carbons and we get from each of the time step. And here I also want to show some of the temporal spatial CO2 field at the top boundary. So just imagine that like there's a Hilliard cells that is put vertically in front of you, but you just see it and at the top of it. So you just stand, imagine like you stand on the top of the Hilliard cells and you look down. And here is the patterns and the morphologies, and you can see around the interface. So please pay attention to like that. The y-axis here is the time, is instead of y anymore. So that is with the time changes, how you can notice about the morphology changes around the interface. So if you still get confused, that was what I'm talking about. Here is the pictures to follow about that. So the A is exactly the screenshot and one-time stop around the interface. So this is the interface. Okay, so we will see like this is the interface. So I took a snapshot from the videos and at one-time step. So this kind of the plot will just corresponds into the lines here. So which is around 45 minutes. So then the horizontal red lines, the dashed lines, what you observe is exactly what you can see on the top of the top boundaries, morphologies. So according to that, we can further investigate some of the instabilities and when the convection happens and where does the plumes migrate and with the times. And here is also some of the results, very preliminary results on the porous mediums. Here the porous mediums is what we constructed by the 3D printer in our laboratories. So this is just a very, very homogeneous porous medium without any of the organics. And then this is also, by this method, we can also construct about its pH evolutions according with the time. Just like that. And then by the same method, we can construct the CO2 concentrations and its total dissolved carbons from that. Yeah, and I think that's all for my presentations, and I put my emails here, and I'm open for any of the discussions and collaborations, and if you are interested. Yes, thank you.
Thank you so much, Yao, for your very, very nice talk. Really, really great experiments. So I'm asking all the people connected online to write their comments on YouTube so we can read them out loud. So if there are no questions for the moment, I can start with a couple of my own. Are there any questions in the studio, maybe? No? Okay, I will start with my own then. So first of all, I had a couple of curiosities about the resolutions of your experiments, like the temporal resolution and spatial resolution that you have in your to show cell experiments.
Yeah. You mean the resolutions? What do you mean?
Which is the temporal resolution and which is the spatial resolution of your data?
Partial resolution?
The spatial, like the size of the pixel that you're using.
Oh, yeah. You mean how large is it, or the centimeters of it, or yeah? So for
how large it is, and yeah, how many points you have in your?
It's 800. It's 8,000 times 4,000 pixels. Yeah.
Okay, and what about time? Like how many images do you take?
It's 10 seconds for each of the pictures, and then we count them together to make the video. Yeah.
Great. That's really cool. And well, we have a question from somebody connected online. Okay, so Jakubis is asking. Well, he's first of all, he's thanking you for the talk, and then he asks you if you can elaborate on why the brightness did not affect the pH value in your calculations.
Yeah, maybe I can share my screen again. I'm sure I have
sure.
Closed it. Very early.
Sure, you can share it again.
Yeah. But can you see my screen now? Okay.
Yes.
Yeah, perfect. So if I return to the things there. So here, so what I mentioned is like, okay, for example, here, if you look at this kind of a coordinate. So for example, there's 2 points, which is A and B. And just imagine that it's located on the line of the OP. So the thing is like, okay, they will have the same length of phi and theta and phi volumes if you want to express it in the polar coordinates, but they will have different radiums. My point is like here, the brightness we're just not influenced by this pH, which means like if your pixel volumes is just located exactly like in the A and B of the OP lines, their pH is exactly the same. So which means like the radiums will not influence it. That is my point of it. I hope that answered your questions, Jakob.
Great. And then I have another curiosity. I mean, if anybody in the studio or online has no other curiosities, I can go on. Oh, Mohammed. Okay.
Yeah.
So when you were talking about like your spatial temporal profiles that you were showing at some point, it was like a very great image with some patterns. And I was wondering if you like looked at some correlations, maybe like at a fixed line in time of this data, or correlation at a fixed time in space or something like that, maybe with some statistical analysis or something like that. I mean, just a curiosity.
It means for these kind of pictures you're talking about?
Yes, for that kind of picture, yeah.
Yeah, it means is there any of the statistics analysis already?
Yeah, like if you, I mean, maybe I'm totally wrong. It's just curiosity that came to my mind. Like if you fix a time instant, maybe, and you're fixing the position along your cell, right? You're looking at everything from the top, right?
Yeah, exactly. Yeah.
So then if you fix the time, you have basically a set of data along along the other sides of your cell, right? Along X, if you're fixing Y. So I was wondering if you like looked at some correlations of these data, like for fixed time along the X profile, or differently if you fix like an X coordinate and you look at the data in time, if you looked at some statistical correlation or something like that. I don't know.
Yeah.
Did you do that maybe?
I haven't done it so far, so this is just a start. But just as I mentioned before, so I would like to. The goal is not only to observe this kind of morphologies and how it migrates. We would like to have more of the quantitative measurements, just like you mentioned for statistics analysis, and also about the Fourier analysis, and to analyze these instabilities by its wave number and wavelengths and so forth. Something like that.
It's great. I mean, since you have like a lot of data basically along both time and space, then maybe it could be an idea.
Yeah, sure, definitely. Yeah. But thanks also very much for mentioning it. Yeah, it's really a good suggestions. Yeah.
And then Mohammed has one question.
Yes, I have one question. Very interesting research for the carbon mineralization or CO2 storage in basalt. As you already know, it's really important for this line of research to know how fast we have this buffering when we have a CO2-rich fluid entering the porous space, which has a, for example, basic pH, and then they mix, and then the pH changes as the fluid advex through the reservoir. Can you share us some insight about how fast is this buffering process, and do you see any effect of fluid mixing, and then what is the difference when you have advection dominated domain or diffusion dominated domain? Can you see any difference in the dynamics?
Yeah, so I would say it really depends on the porous mediums and its complexity. I think it will definitely influence a lot about the whole of the pictures. But the thing is, it's like here, so we only did the experiments for the heliotrope cells, and so you can notice like it has a small dimensions, which is 32 centimeters to 16 centimeters of such dimensions. But for such a small dimensions, you can notice like actually the CO2 will just change the pH very fast, as you mentioned, as you can see from here. For the several 10 minutes, you can notice like it can also change a lot of the pH from that, and just by a little bit about the CO2 dissolved. As you can see, the total amount of the CO2 that get dissolved in such a small dimensions actually is not very high. It's something like 10 to -5 moles. That density can cause such an effect. So I would say like for such of the dynamics, I think there will be many of the things we'll just to control it. The heterogeneity of your porous mediums, and also say the porosity, and also and so on and so on. and even the rock types, I would say, for different minerals, I think it will also have the influence for the whole picture. I think it's challenging to say at the moment, but that is but I'm still on the way and just explore more of that. And now I'm still trying to use the heritage cells, but I would also want to use some of the other more complex and the porous mediums. So now I'm using the 3D printed models, and maybe afterwards, if we get more known about that, and we can proceed to some of the real rocks and try different things. And then maybe we can see more of the interesting stuff, I would say.
Excellent. Thank you very much.
Thank you.
Great. So maybe when you have more results with the 3D printed models or everything, you can come back and give another talk here because it's really, really cool work.
Yeah. Thanks so much.
Okay, so I think that if we have no more questions from people connected online or from the studio, I think we can end this session for today here. With this, I thank again our speaker for today, and I thank again all my friends and all the people from the PMTTT Talks team for organizing this kind of event. So our next talk is scheduled for the 11th of June. We will have two speakers again. One will be Riccardo Sprocati from NTNU, and then we will have Negar Nasri from Harvard University. Riccardo will talk about enhancing solid transport through electrokinetics, while Negar will focus on how hydrogen is produced in basalt rocks in response to hydrothermal reactions. So I remind you that in case you want to take part to this event, you want to present your work, you can just contact us at the email that you see here on this slide. So with this, I thank you all again and see you next time at the next PMTTT talk. See you. Bye.